You know, I was spending some time this week looking at the photos in that stack of research you sent over.
Oh, yeah.
And if you just glance at them, these high-resolution fluorescent microscope images, you'd think you were looking at a masterpiece.
Right. They're beautiful.
They're gorgeous. You see these glowing layers of neurons, complex cortical structures, and everything looks absolutely pristine. It looks perfect.
It is visually incredibly deceptive, isn't it?
It is completely deceptive because you look at these cerebral organoids, these so-called mini brains grown from stem cells, and the headline is always the same. It's always, "Scientists grow human brain in a dish."
That's the soundbite, yeah.
It sounds like we've solved it, like we have a perfect model brain sitting on a lab bench. But then I started digging into the actual, the methods sections of these papers, the fine print.
hmm
And I found this term that just keeps popping up. It's almost like a dirty little secret the field has been trying to hide.
I think I know exactly what you're talking about. The physical limit.
The necrotic core.
Yeah. That's it. It is the elephant in the room. Or the petri dish, I guess.
It sounds like a heavy metal band, but it's actually a biological failure, right? We grow these things, they look perfect on the outside, but once they get to be about the size of a lentil, maybe 4 millimeters across,
That's the wall.
the center just rots.
It rots. That is the blunt reality of it. And it's strictly a physics problem. It's not that the biology is wrong. It's that we just run headfirst into the limits of diffusion.
Diffusion, meaning just things seeping in from the outside.
Exactly. When you have a cluster of cells floating in a nutrient bath, the oxygen and the sugar can only seep in so far, roughly 2 millimeters, maybe 4 if you're lucky.
Mm.
Beyond that, the cells in the center are suffocating.
So they're starving.
They're starving, they're choking, and they die. And you get this hollow, dead center surrounded by a shell of perfectly healthy-looking neurons.
Which is a massive problem if you're trying to model a human brain. Because, last I checked, my brain isn't hollow. And surely having a rotting center affects the healthy parts too.
Oh, absolutely. It's not just about the dead cells effectively doing nothing. It's about what that death does to living cells. When cells die via necrosis, they don't go quietly.
They don't just fade away.
No, they burst, they rupture, they release toxins, inflammatory signals, all this cellular debris. So even that healthy outer shell, it's constantly fighting a war against the toxic waste dump sitting right in its own core.
So for years, we've been trying to study complex brain diseases using a model that is inherently stressed out and dying from the inside.
It's a huge confounder. You never know if what you're seeing is the disease you're studying or just the organoid reacting to its own toxic center.
So that sets the stakes for this deep dive. We aren't just talking about making them bigger for the sake of size. We're talking about the quest to solve that fundamental physics problem, to vascularize the mini brain.
Right. We have to build the plumbing. We have to install the circulatory system.
And reading through these papers on bioengineering, genetic hacking, even, transplanting human tissue into mice, it became really clear that this isn't just a plumbing job. It's not just installing pipes to bring in water and take out the trash.
No, it's much more profound than that.
The sources you shared make a really compelling argument that the blood vessels are actually architects.
That is the key insight. The vessels tell the brain how to build itself. They're not passive infrastructure. They are active construction partners.
So today we're going to look at how scientists are fusing organoids together like Lego blocks, how they're using these internal hacks to flip genetic switches, and the ethical weirdness of stroke in a dish models. But before we get to the engineering, I want to go back.
Okay.
I want to go back to the womb. Because I think I misunderstood something really fundamental about how we develop.
What do you mean by that?
Well, I always assume the brain grows, right? It lays down all the neurons, all the layers, and then the blood vessels just kind of fill in the gaps. Like you build a house, and then the plumber comes in afterwards and routes the pipes through the walls you've already built. But the paper on Nature's Blueprint suggests that's just completely wrong.
It is completely backwards. In the embryo, the nervous system and the vascular system are what we call parallel travelers. They're, childhood friends. They grow up together, literally hand in hand. You cannot have one without the other.
So they're co-developing from day one.
From the earliest moments.
Walk me through that timeline. Because in those first few weeks of an embryo, we're talking gestational, weeks 4 and 5, we are tiny. We're basically a smudge of cells. We don't have a heart pumping blood to the brain yet, do we?
Not effectively, no. At that stage, weeks 4 and 5, the neural tube, which is going to become your brain and spine, is still open. It's very thin. So diffusion actually works fine here.
Just like in the dish.
Exactly. It's soaking in amniotic fluid like a sponge. The distances are microscopic, so oxygen and nutrients can just drift in. No problem.
Okay, so nature starts with the Petri dish method. Just let it soak.
Exactly. But nature knows that won't last. Around gestational week 7, things get critical. The brain tissue starts to thicken. It starts layering up. Suddenly, those walls are way too thick for diffusion to work. This is the tipping point.
This is where you'd get a necrotic core in the embryo.
You absolutely would.
Yeah.
If blood vessels don't show up right at that moment, the tissue dies. It's a developmental emergency.
So what happens inside the developing fetus at that point? What's the rescue mission?
You get the formation of what's called the perineural vascular plexus.
Which is a mesh.
Think of it like a fine, delicate scaffolding net made of blood vessels that wraps around the entire outside of the neural tube.
Okay, so a cage of vessels.
A cage, yeah. And then in a process called angiogenesis, those vessels start to invade. They literally burrow from that outside mesh deep into the brain tissue, heading toward the center, toward the ventricular zone.
The ventricular zone, that's the neuron factory, right? That's where all the action is.
Correct. That's where the neural stem cells are churning out baby neurons. And here's where that architect concept really comes into play. Those blood vessels aren't just wandering randomly. They are following signals from the brain's own cells, the glia cells.
So the brain is biting them in.
Yes, the radioglias are like air traffic controllers, laying down the flight paths. But, and this is the really cool part, the vessels are also shouting orders back to the neurons.
It's a two-way conversation.
A very loud one. The endothelial cells that make up the blood vessels, they secrete chemicals. Factors like GABA, which we think of as a neurotransmitter later on, but here it acts as a stop signal. And another one called VEGF. These chemicals tell the neurons, okay, stop dividing now. Time to pack your bags and migrate. Go that way.
Wait, so if I'm growing a mini brain in a lab and I don't have blood vessels, I'm not just starving the cells in the middle, I'm missing the foreman on the construction site.
Precisely. You're missing the instructions. Without the vasculature, the neurons don't know when to stop dividing. They don't migrate to the correct cortical layers as efficiently. So you end up with a brain that isn't just small and dying, it's disorganized.
It's structurally wrong.
It's structurally wrong. It's like trying to build a skyscraper where the steel workers and the electricians and the drywallers aren't talking to each other. You get a building, but it's not going to be functional.
That explains so much. It explains why the non-vascularized organoids we've had for the last 10 years are kind of messy. They have neurons, sure, but they don't have that beautiful 6-layer architecture of a real human cortex.
They're confused. They're doing their best with half the blueprint, but they're missing these crucial developmental cues.
Okay, so we know we need the vessels. We know where. Now we get to the really hard part, the engineering.
Right. How do you actually do it?
And I have to say, the how-to section of this research read like a comedy of errors. Because apparently brain cells and blood vessel cells, they hate each other in a dish.
Hate might be a strong word, but they are definitely socially incompatible in the lab.
Why? They coexist perfectly fine in my head right now.
In your head, yes, because the body is an incredibly complex and buffered system. But you have to remember where they come from. In the embryo, brain cells arise from the ectoderm. That's the outer germ layer. Blood vessels, on the other hand, come from the mesoderm, the middle layer.
So they're from different tribes, biologically.
Very distant cousins, let's say. And in the lab, to keep cells happy, we feed them media, which is basically a nutrient broth. But the broth that makes a neuron happy full of specific neurogrowth factors is often toxic or just completely useless for an endothelial cell, which makes up the blood vessels.
And vice versa, I assume?
And vice versa.
Yeah.
The stuff that makes endothelial cells thrive can actually inhibit neurons from developing properly. So if you just throw them in the same bucket, one of them usually suffers, or both of them do.
So the challenge isn't just put them together, it's find a diplomatic solution where they don't poison each other.
Exactly. It's a resource management problem on a microscopic scale.
Let's look at the solutions then, because the research lays out about 5 different major strategies. And I want to start with the most obvious one, which they call co-culture, or the mixing method.
Strategy A, the brute force approach.
Strategy A, throw them in the blender. Basically, you grow your brain organoid, and then you just dump in some blood vessel cells, usually HUVECs.
HUVCs, yes. Human umbilical vein endothelial cells. They are the lab rats of the vascular world. They're robust, they grow fast, they're easy to get.
So you dump these HUVECs into the dish with the brain organoid and just hope for the best.
You cross your fingers and hope for the best, and sort of it works. I mean, the endothelial cells are genetically programmed to make tubes, so they do. They self-assemble and form these little capillary-like structures inside the organoid.
I sense a big butt coming.
Oh, it's chaotic.
The paper described it as random and disorganized.
That's putting it mildly. In a real brain, you have this beautiful hierarchy. Big arteries branch into smaller arterioles, which branch into tiny capillaries. It's a tree. In the co-culture method, it's more like a pile of dropped spaghetti.
Huh. Okay.
You get tubes, but they aren't connected effectively. They don't have a clear in and out. They don't penetrate deep enough to actually solve that necrotic core problem we started with. So you still get that dead center.
So it's cosmetic vascularization. It looks like it has vessels if you squint, but it doesn't function like a real circulatory system.
It was a start, a proof of principle. But it was definitely not the final solution.
Okay, so mixing didn't work great. Let's talk about strategy B, which I found much, much cooler. The internal hack. This involves genetic engineering.
This is where things get really clever. Instead of trying to force two different cell types to live together, the question became, what if we could convince the brain organoid to make its own blood vessels?
Like reprogramming the cells from the inside out.
Exactly. There's a specific gene, a transcription factor called ETV2.
And transcription factors are like the master switches of our DNA, right? They turn whole sets of other genes on or off.
Precisely. They are the generals giving the orders. And ETV2 is the master switch for a blood vessel cell. If you turn on ETV2, a stem cell is pretty much committed to that fate.
So what did the researchers do?
Researchers, and this was highlighted in the Keiko study in your notes, took the stem cells that were destined to become the brain organoid, and they engineered a small percentage of them, maybe 5 or 10%, to express this ETV2 switch.
So you have a ball of cells, and 95% of them are saying, I'm going to be a brain, but 5% contain this sleeper agent code.
A perfect analogy. And when you're ready, you flip the switch, usually by adding a specific chemical trigger to the media. And that 5% wakes up and says, actually, I'm not a neuron. My orders have changed. I'm an endothelial cell now. And they start forming tubes right there inside the developing tissue.
That is incredible. So it's self-assembling plumbing, built from within.
It's intrinsic vascularization. And the results were drastically better than the mixing method. Because the vessels were forming alongside the neurons, from the very beginning, they integrated so much better. They followed the natural contours of the tissue.
And the necrotic core.
It's significantly smaller. And interestingly, the neurons around these vessels matured faster. They were firing electrical signals earlier and more robustly than in the non-hacked organoids.
Because the foreman was there from day one.
Because the foreman was there.
Yeah.
Even though there was no blood flowing, let's be clear, there's no heart pumping here. Just the presence of the vessel cells provided those essential chemical signals, those architectural cues we talked about.
But you just hit on the limitation. There's no pump.
Right. You have an excellent network of pipes, beautifully laid out, but they aren't connected to the city water main. So you still have a flow problem. You solve the architectural issue, but not the perfusion issue.
Which brings us to strategy C. This one is my favorite because it just feels like playing with biological Legos, the assembloids.
Assembloids are the most modular approach and probably most elegant right now. The idea is, biology's hard when you mix things, so let's simplify. Let's grow a perfect brain organoid in dish A. And let's grow a perfect vessel organoid, just a ball of pure blood vessel cells in dish B.
So you get them both healthy and strong separately in their own ideal environments.
Right. No fighting over the food media, no cross-contamination of signals. Then, when they are both mature and ready, you take them out and you place them right next to each other in a little droplet of gel.
And they just fuse.
They fuse. And what happens next is fascinating because it mimics nature so perfectly. The vessel organoid wraps itself around the brain organoid, almost like a hug.
Wow.
And then, just like in that embryonic process we talked about, the vessels start to sprout and invade the brain tissue from the outside in.
It recreates that nature's blueprint from gestational week 7. It's an engineered recapitulation of development.
Exactly. It respects the developmental timeline. And the Sun et al. Study from 2022 showed something else with this method that was a total surprise. A buy one, get one free deal. biologically speaking.
This was the microglia.
The microglia, these are the resident immune cells of the brain. They're the brain's janitors, its police force, its gardeners. They are absolutely critical for a healthy brain.
And usually our mini brains don't have them at all.
Never. Because again, microglia come from the mesoderm, not the ectoderm. They're outsiders that migrate into the brain during development. So our standard brain organoids have no immune system.
Which is a huge problem if you want to study, say, Alzheimer's or multiple sclerosis.
Exactly. But here's the kicker. When they made the vessel organoid, because it came from that mesoderm lineage, it naturally produced microglia precursors. So when the two organoids fused, the microglia migrated into the brain tissue right alongside the invading blood vessels.
So suddenly you have a brain model that has neurons, blood vessels, A&D, a functional immune system.
Yes. For the first time in a dish. And that is a monumental game changer for studying diseases. Because so many brain diseases, Alzheimer's, autism, schizophrenia, have a huge neuroinflammatory component. You can't study inflammation if you don't have the cells that cause inflammation.
I want to pause on the microglia for a second because there was a detail in that study that just blew my mind. They saw the microglia pruning synapses. What does that even mean?
It's one of the most important jobs microglia have. You know how when you learn something, your brain is making new connections, new synapses?
Right.
Well, a big part of learning and development is also breaking connections. Your brain overwires itself initially, especially when you're young. To be efficient, you need to cut out the noise. You need to snip the weak or unnecessary connections. That's called synaptic pruning.
And the microglia are the gardeners with the shears.
They're literally the gardeners. They go around and they eat the bits of synapse that aren't being used or aren't strong enough. And in the assemblage study, they used electron microscopy and found microglia with bits of synaptic material inside their cellular bellies.
They had evidence of the meal?
They had the receipts. This implies the organoid wasn't just a static ball of cells, it was actively refining itself. It was undergoing a process that is essential for learning, memory, and cognition in a real brain.
That is spooky and amazing all at once. But we still have that one lingering problem. We have pipes, we have immune cells, we have structure, but we still don't have a heart. We don't have real blood pressure, real flow.
And for that, you have to leave the Petri dish entirely. For that, you have to go to strategy D, the host method.
This is where we get into the Frankenstein territory, transplanting human mini-brains into the brains of mice.
It sounds like pure sci-fi, but it's the gold standard for achieving true vascularization right now. You take your human cortical organoid and you surgically implant it into a mouse's brain, usually replacing a small piece of its cortex.
So you're putting a human brain patch onto a living mouse brain.
You are. And then biology just takes over. The mouse's circulatory system recognizes this new tissue and says, hey, this needs oxygen. And the mouse's own blood vessels grow right into the human tissue. They anastomose, they connect with the human capillaries that might have started to form.
So now you have human neurons being fed by mouse blood, pumped by a mouse heart.
Exactly. And because you have real blood flow, real perfusion, the human tissue thrives. It grows much bigger than the lentil limit. The neurons become incredibly mature, forming complex synapses and networks that you just don't see in a dish.
But now you have a chimera, a human-mouse hybrid brain, in a sense.
You have a chimera, and this brings up a host of practical issues. You know, you can't use this for high-speed drug testing because surgery on mice is slow and expensive, and of course some very significant ethical issues.
We will definitely circle back to the ethics, but before we do, there's one last engineering strategy for the people who don't want to do surgery on mice.
Right. Strategy E, the chip method, organ on a chip.
This feels like the pure bioengineering solution.
It is. Instead of a dish, you grow your organoid or your vessel cells on a small glass or polymer slide, a microfluidic device that has tiny channels the width of a human hair etched into it.
And you hook this up to a pump.
You hook it up to a precise pump that pushes nutrient fluid through those channels to mimic blood flow.
And it's not just about delivering the food, right? The reading you sent mentioned something called shear stress.
This is a physics concept that turns out to be biologically vital. Shear stress is the friction, the dragging force, of fluid moving across the surface of a cell. And the endothelial cells that line your blood vessels are designed to feel that flow.
So they can tell blood is moving.
They can tell, and it changes their behavior. When they feel that friction, they align themselves with the direction of the flow. They stretch out, and most importantly, they lock together much tighter.
They stiffen up.
They mature. They activate the genes for those tight junctions we'll talk about. Without that flow, without that shear stress, the vessels tend to be leaky. They're like loose, drippy garden hoses. But put them on a chip with a pump, and they form watertight seals.
And that watertight seal is the perfect transition to the next big topic. Because in the brain, Watertown is the difference between life and death. We need to talk about the blood-brain barrier.
The BBB, the holy grail of drug development and the brain's ultimate security system.
So we've built the vessels, we've used chips or mice or genetic hacks or assembloids. Now the big question is, do they actually work? Do they act like the bouncer at the club, keeping all the bad stuff out?
This is what distinguishes a truly functional, vascularized organoid from just an organoid with some tubes in it. The blood-brain barrier is a highly specialized structure. The endothelial cells in your brain are stitched together with proteins called tight junctions.
The sources mention specific markers for this, like Claudin 5.
Yes, Claudin 5 and ZO1. Think of these proteins as the teeth of a zipper. They physically zip the cells together so tightly that almost nothing can squeeze between them. Not bacteria, not toxins, and unfortunately for medicine, not most drugs.
And they also found pumps, right? A specific one called P-glycoprotein.
The P-glycoprotein pump. It's fascinating. It's an active transporter. It sits on the vessel wall, and it's like a bouncer with an ejector button. If a foreign molecule, like a drug, manages to sneak past the zipper and get into the cell,
This pump just grabs it and throws it back out.
it literally grabs it and spits it back out into the bloodstream. It's one of the major reasons why treating brain cancer is so incredibly hard. The tumor is riddled with these pumps, and it just spits the chemotherapy right back out.
So in these new vascularized organoids, specifically the ones from the Sun and Kakir studies, did they find these zippers and pumps?
They did. Using genetic sequencing and protein staining, they found high expression of Claudin 5, they found ZO1, and they found functional P-glycoprotein.
Functional is the key word there, isn't it?
It's everything. It means you can test it. They could add a dye that is a known target of the pump and watch under the microscope as the organoid's vessels actively pumped it out. It proves the barrier isn't just there, it's working.
So this means we can finally test drugs in a dish and get a realistic answer. Before, if you just dumped a drug on a naked organoid...
It would obviously work because there was no barrier to cross.
Yeah.
Then you'd get excited, you'd try it in a human, and it would fail spectacularly because it couldn't get into the brain.
So now we can ask the right questions in the dish. Can this molecule actually unzip the zipper? Can it disguise itself to evade the pump?
Precisely. It makes preclinical drug screening infinitely more predictive and powerful.
This moves us into the realm of what the papers call the neurovascular unit. It's not just the barrier, it's the whole neighborhood of cells working together.
Right, the NVU. It's the neuron, the blood vessel, the pericyte, which is a cell that wraps around the vessel to strengthen it, and the astrocyte, which acts as the middleman with one arm on the neuron and one on the vessel.
The whole support crew.
The whole support crew. And what we're seeing in these advanced vascularized models is that the presence of this full unit actually changes the neuron's behavior. The neurons in these VBO's vascularized brain organoids show much more complex electrical activity. They have action potentials that look more like an adult brain than a fetal one.
So the plumbing doesn't just keep them alive, it helps them grow up.
It creates the microenvironment that is necessary for them to mature properly. It's absolutely critical.
This feels like the right moment to pivot to the so what question. We have these incredible complex models. We have the zippers, the pumps, the immune cells. How does this actually help us understand when things go wrong in the human brain? The section on disease modeling was full of surprises for me, especially regarding schizophrenia.
That is one of the most compelling recent findings. Historically, we have looked at schizophrenia as a disease of the synapse, imbalanced dopamine, misfiring neurons. A software problem, essentially.
Right, a neurochemical issue.
But the vascularized organoid studies are suggesting it might be a hardware problem, too. A plumbing problem.
There was that specific study where they took cells from patients with schizophrenia, made these vascularized organoids, and compared them to organoids from healthy controls. And the result was completely counterintuitive.
It was. You might expect the diseased organoids to have fewer blood vessels or weaker ones, but they actually had more, a higher density of vascular networks.
Which on paper sounds good. More blood, more oxygen, right?
You'd think so. But quantity does not equal quality. When they looked closer, the vessels were disorganized and, crucially, they were inflamed and leaky. They didn't express enough of those tight junction proteins, the zippers we just talked about.
So the blood-brain barrier was down.
The barrier was compromised. It was permeable. This suggests that the brain tissue in these patients might be constantly exposed to inflammatory molecules and other signals from the blood that just shouldn't be there. It changes the whole paradigm.
It reframes the disease.
Completely. Maybe we shouldn't just be developing drugs that target dopamine receptors in the neurons. Maybe we should be developing drugs that treat the blood vessels and seal the leak.
You could never, ever have seen that in a standard Petri dish with just neurons. You'd miss the potential root cause entirely.
You'd see the fire, but you wouldn't see the arsonist who snuck in through the broken window.
Then there's autism spectrum disorder, or ASD. How does vascularization help us understand that?
ASD is often a story of asynchronous development. The timing of brain construction is off. In a typical brain, you need a very precise balance between excitatory neurons, which are like the gas pedal, and inhibitory neurons, the brake. In many forms of ASD, that balance is skewed.
And since the blood vessels are the architects or the foreman...
Exactly. If the foreman is late to the job site or gives the wrong signals at a critical time, the scaffolding for the whole building gets built wrong. These vascular studies are helping us see how early disruptions in blood flow or vascular signaling might lead to that cortical layering being miswired from the very beginning.
So we can trace the origins of the disorder back to the womb.
We can create a developmental timeline and pinpoint where things might have first gone awry.
Let's talk about something more acute. Stroke. I mentioned the idea of a stroke in a dish in the intro. It sounds like a paradox. How do you give a stroke to something that doesn't have a body?
Well, at its core, a stroke is ischemia, a sudden, severe lack of oxygen and nutrients because a blood vessel is blocked. In these vascularized models, especially the ones on microfluidic chips, you can simulate this perfectly.
You just turn off the pump?
You turn off the pump. Or you replace the oxygen-rich media with oxygen-deprived media. And you can watch the fallout happen in real time.
And why is that better than, say, a mouse model? We've been giving mice strokes in the lab for 50 years.
We have. And we have cured stroke in mice hundreds of times over. But almost none of those drugs work in humans.
The famous translational gap.
It's a chasm. The mouse brain is different. Its metabolism is different. Its tolerance for lack of oxygen is different. Its immune response is different. The human organoid offers a better platform. Specifically, it lets us see the penumbra.
The penumbra, what's that?
When a stroke hits, there's the core, the zone that dies almost immediately. It's gone. But around it is a larger area called the penumbra. This is tissue that is stunned, damaged, but still alive. It's hanging on by a thread. That is the tissue we are racing against the clock to save in the ER.
So that's the therapeutic target.
That's the whole game. In these organoids, we can watch the penumbra in real time under a microscope. We can see how the human microglia respond. Do they help clean up debris, or do they go on a rampage and attack the damaged but living tissue? We can test drugs specifically designed to save those penumbral human neurons.
It's amazing to think we can watch that life or death battle happen on a slide. And speaking of battles, we have to talk about cancer, specifically glioblastoma.
The most aggressive and devastating primary brain tumor. And it is essentially a vascular vampire. It cannot survive without hijacking the brain's blood supply.
It recruits blood vessels to feed itself.
It does. It's a process called tumor angiogenesis. Glioblastomas are masters at secreting chemicals that force new blood vessels to grow right into the tumor mass.
So with these models, we can see how the tumor hijacks the pipes.
Yes. And even better, we can test drugs that try to stop it. Yeah. But this leads us to the really futuristic concept of the avatar.
This is the personalized medicine angle. This is where it gets really powerful.
Right. Imagine you have a patient, let's call her Jane, with a glioblastoma. We can take a small sample of her skin cells, reprogram them into stem cells, iPSCs, and grow a vascularized brain organoid that is genetically Jane.
It's her mini brain.
It's her mini brain. Then we take cells from her actual tumor biopsy and we seed them into her organoid. We effectively create an avatar of her brain and her specific cancer living in a dish.
So instead of giving Jane chemotherapy and just hoping it works,
we can test 20 different chemotherapy cocktails on dozens of her avatars first. We can see which drug actually crosses her specific blood-brain barrier. We can see which one kills the tumor cells, but spares her healthy neurons.
You do the trial and error in the plastic dish, not in the patient's body.
Exactly. It saves precious time, which is the one resource glioblastoma patients have very little of, and it spares them from the brutal side effects of toxic treatments that were never going to work for them in the first place.
This brings us to a major benefit of this technology that we touched on in the outline. Reducing the use of animals in research. The 3 Rs.
Replacement, reduction, and refinement. That is the ethical framework that guides all animal research.
We rely so heavily on mice. But as you said with the stroke example, a mouse is not just a tiny human.
It's really not. A mouse brain is smooth. It doesn't have the deep folds, the gyri and sulci, that give the human cortex its massive surface area. It lacks certain cell types that are specific to primates. And the gene expression profiles for thousands of genes are completely different. This is a huge reason for translational failure.
So these vascularized organoids act as a bridge over that translational gap.
They act as a better filter. The idea isn't to replace animal models entirely, not yet. But you can use the organoids for high-throughput screening first. If a drug works in a mouse A and D, in a vascularized human organoid, you suddenly have much, much higher confidence that it will work in a person.
So we can screen out the obvious failures earlier.
And that means we can drastically reduce the number of animals that are used for testing drugs that were doomed from the start. It's a win for science and a win for animal welfare.
But, and there is always a but, when we get this advanced, we have to talk about the ethical implications of the organoids themselves. Because we are making these things more and more complex. We are giving them blood vessels. We are making them live longer, potentially for years. We are seeing complex, synchronized electrical activity.
This is the consciousness debate.
It's getting louder in the field, isn't it?
It is, and it should be.
Mm-hmm.
As we solve that necrotic core problem, these things get bigger, the neural networks get denser and more interconnected, and we are starting to see synchronized electrical waves, oscillations, that look suspiciously like the EEG patterns of a preterm infant.
That phrase alone just gives me chills.
It should. It demands that we treat this work with the utmost respect. Now, the current consensus among most bioethicists and scientists is that we are still very far from sentience or consciousness.
What's the main argument for that?
The main argument is the lack of sensory input. These organoids are locked in a dark, silent box. They can't see, hear, or feel. They are processing signals in a complete void. It's a brain in a vat, without the vat being connected to anything.
But we are working on that too. I read about researchers hooking up retinal organoids, little eye cups that can detect light-to-brain organoids.
We are. And that's where the ethical line starts to get really blurry. If you give it input, if you give it an optical sensor that translates light into neural signals, and the brain organoid processes that input, at what point does neural activity become experience?
At what point does a reaction become a perception?
Exactly. We don't have a scientific definition for that yet, let alone a philosophical one.
And we don't really have regulations for it either, do we?
Not specific ones. There are broad guidelines for stem cell research, but nothing that says you can only grow an organoid for X number of months. Or, if it displays Y type of brainwave, you must terminate the experiment. We need to have that conversation now before the technology outpaces our ethical framework.
And we need to define what suffering might mean at a cellular level.
Right. If an organoid has a functional pain pathway, can it feel pain in a dish? It's a question that sounds like science fiction, but it's becoming a real consideration.
And then there's the chimera issue we touched on with the mouse transplants.
That is another very sensitive area. When you transplant a significant amount of human brain tissue into a mouse, you create a human-animal chimera. And the immediate public fear is, does that mouse become smarter? Does it become humanized?
Does it start doing algebra in its cage and asking for a lawyer?
The evidence so far is a firm no. The human cells integrate, they fire, they connect with the mouse cells, but they don't seem to fundamentally restructure the mouse's brain architecture into a human one. The mice is still a mouse. But it raises deep philosophical questions about human dignity and species boundaries.
About what it means to be human.
Exactly. And then there are the more practical, legal issues. Who owns these avatars? If you grow an organoid from my skin cells and it's used to discover $1 billion drug, do I get a cut? Is that my intellectual property?
Yeah. And informed consent.
Informed consent is a huge frontier here. Most people who donate cells for research sign a form. They imagine a scientist looking at their cells on a slide for an hour. They don't imagine their cells will be built into a living, functioning mini-brain that might live in an incubator for 3 years, potentially being part of a neural network study that borders on cognitive science.
We need to be transparent with donors about just how alive these models are becoming.
Transparency and public dialogue are the only ways this field moves forward without a major public backlash.
It is an incredible amount to process. We've gone from a lentil-sized blob with a rotting center to a vascularized, perfused, complex tissue that mimics the architecture of the human brain, capable of modeling schizophrenia and strokes and potentially challenging our very definitions of life.
It is a remarkable trajectory in just over a decade. Vascularization really is the missing link. It's the key that transforms a simple cluster of cells into a true high-fidelity tissue model. It brings oxygen, structure, immune defense, and a level of complexity we could only dream of before.
So, what is the next frontier? If you look ahead 5 or 10 years, where is this all going?
I think we're moving toward the body on a chip. Not just a brain organoid in isolation, but a brain organoid connected via microfluidic vessels to a liver organoid, a heart organoid, and a kidney organoid.
A closed-loop system, a mini-human on a slide.
Exactly. So you can introduce a drug and see the whole picture. You can ask, does it cure the brain but damage the liver? Does the liver metabolize the drug into something that then becomes toxic to the heart? It's about modeling systemic effects in a human-relevant system. That is the dream.
That's incredible. I want to leave our listeners with a thought that came up while I was reading about that necrotic core again. We view that dead center as a failure, a flaw in the model that we have to fix.
Of course. It's a technical problem to be solved.
Right. But hearing you talk about the ethics, about consciousness, I started to wonder,
I think I know where you were going with this.
if vascularization allows these models to survive for years, to grow larger and more complex, and we start integrating sensory inputs, are we approaching a future where the model is no longer just a simulation, but a distinct biological entity with its own rudimentary experience?
It's the question we have to grapple with.
And if so, does that necrotic core, that simple physical limitation that stops them from growing, actually serve as a convenient, built-in ethical failsafe that we are currently working very, very hard to remove?
That is the ultimate and perhaps unsettling question. By solving the biological problem, are we inadvertently creating a massive ethical one that we aren't prepared to handle?
Something to mull over. Thank you so much for joining us on this deep dive into the vascularized brain.
My pleasure. It was a fascinating discussion.
And to our listeners, keep curious, keep asking the hard questions, and we'll see you in the next deep dive.